culturing the plant parts. Based on this totipotency character, we can culture the plant parts and tissues to propagate, to develop a new plants. So this altogether called as plant tissue culture. First, tissue means the group of cells called as a tissue. Because of the plant's totipotency property, we can culturing the plants. So there are some requirement to culturing the plants, such as the main requirements. We need culture media, and also we need aseptic condition, and also one proper laboratory. These are requirement. First of all, in general, how the plants are obtained. In general, how the plants are obtained. The plants are obtained, plants are developed, or plants are germinated in a process of life cycle. First, in life cycle, the seed germinate to produce the plant. After production of plants develop say plant pods, root, shoot, and leaves. When reach maturity stage, which develop the flower pods, which act as a reproductive. After formation of reproductive organs, fertilization takes place. As a result, formation of zygote, which developed embryo, and finally seed. This is the process generally occurs in plants. For taking this all process, here the plants require the more time. One plants to produce another plants, the based on the species gets for years, takes for years. But the growing population in a world, we need the plants requirements. That's why we should get the number of plants within time by tissue culture process. So let's see the tissue culture. First of all, requirement of tissue culture. Already told you, in general plants developed, plants develops by seed which is germinated on soil. So in general soil requirement. So in the place of soil, here, just wait. In place of soil, here the two fixation required culture media. Generally, we use agar medium. It is act as a soil. That means to fixation of the plant or fixation of any explant. So agar, that is the culture media. After culture media, we should provide aseptic condition and proper laboratory. First, how to prepare the culture media. The media on which the explant is cultured called culture media. Actually, I will tell you what is explant later. If you take explant, culture the explant on media, that is called culture media. The culture media composed of various nutrients required for proper culture. What are the requirement to plant to grow? In general, for plant to grow, most of the micro and macronutrients and also phytohormones, they are all required. Already already we learnt in practical in practical training classes. In practical training classes already we have seen the preparation of culture media. What are the requirement involved in it? We prepared stock solutions also. So like that, just can recalled once again here. So like in general nutrients, here in tissue culture also to prepare media require some nutrients, macronutrients and micronutrients. Such for example, micronutrients are iron, manganese, zinc, molybdenum, copper, boron, as well macronutrients such as nitrogen, sulfur, phosphorus, potassium, calcium, and magnesium. These all ingredients we have taken during practical training classes to prepare culture media. Already most of you must have learned how to prepare culture media. So this is the media preparation. What happened during the preparation of media? In media, we utilize the most of the nutrients, right? So due to the nutrients such as glucose also required, proteins also required, vitamins also required, like vitamin like thiamine, pyridoxine, nicotinic acid, some amino acid, arginine, asparagine, inositol. So this presence of these all nutrients and amino acids, there is a chance to infect with the microorganisms because of some nutrients. That's why we should do sterilization of the culture media. The sterilization process called as aseptic condition. That means we provide the aseptic condition. Aseptic condition. After aseptic condition provide, maintain the sterilized laboratory. What are present in tissue culture laboratory mainly? Already told you during practical training classes. Autoclave. What is the function of autoclave? After preparation media, after preparation media, culture media, most probably we prepared MS medium. After preparation MS culture media, these culture media keep inside the autoclave to sterilize. So as to sterilized to provide aseptic condition. After sterilization, what what will do after sterilization? This sterilized medium inoculated into the laminar airflow. These are the main two instruments in tissue culture laboratory. One is autoclave. The autoclave sterilized the medium, prepared medium. After sterilized, inoculated into the inoculate and incubate inside the laminar airflow. Already told you about the laminar airflow. Laminar airflow, which is produce the UV light and temperature to kills the microbes. If during the inoculation and incubation time, if any microbes, if there is a chance to attack any microbes, but this laminar airflow with help of UV and with help of temperature and light kills the microbes. Do not contaminate the culture media. So these are the uh, basic requirement of plant tissue culture. So first culture media preparation already we have learnt in training classes and theory also today, and aseptic condition to proper aseptic condition by sterilizing with help of autoclave and also laboratory organization. That means we should properly use autoclave and also laminar airflow, right? So this is about requirements. Just Right. So let's see the techniques in tissue culture. First of all, in general, basically some steps main generally in first steps we want explant. Before the explant, as I told earlier, prepare culture media, sterilize culture media. Number two. Number three, select explant, sterilize explant, inoculate explant into laminar airflow, incubation in laminar airflow. After incubation, production of plantlets. Finally, transfer the plantlets to outfield. So this is the in general steps. Let's see in proper way. First of all, actually not given in these steps. First, we already prepared MS media. Already we know how to prepare MS media. After preparing MS media, we select the explant. What is explant? Explant means any part of the plant. Any part of the plant called as a explant, such as leaf, root, stem, or tissue parts also, or even cell, pollen grain, ovule. These are all. Anyone act as an explant. First of all, we select a desired plant because you want to get some desired characters in next coming generation. So before that, you have to select desired characters plant. So that is explant. After that is called explant, intact plants such as meaning explant. After selecting explant, because we should sterilize the explant. Sterilize explant. Why we do the sterilization? Because actually explant is present in outfield. In outside the weather, there are number of microbes. So if do not sterilization, you directly inoculated into the media. What happened? The entire media, there is a chance to contaminate. That's why, so as to avoid this contamination, first after selected explant, you should remove. So you should sterilization. You should sterilize the explant. A suitable explant is selected and then it's sterilized. The sterilization main chemical with help of HgCl2. With help of HgCl2 and some detergents, we should sterilize the explant. After that, proper trimming of explant. After sterilization, actually, we need to trim the explant. And again sterilization. Again sterilization. Here sterilization glassware also. That mean before the preparation of media, actually media preparation inside the test tube, right? So the test tube also sterilized. Explant washed and sterilized distilled water before inoculation. First you selected, after selected, you remove the dust or any other extra particle you removed. Trim the proper way. Surface sterilization with help of HgCl2 and also at the same time sterilization of glassware also. And finally, before inoculation, sterilization with distilled water. And already told you culture medium sterilized in autoclave. Culture media sterilized autoclave. After that, inoculation of explant in the culture media under the aseptic condition. Under aseptic condition, the aseptic condition means sterilization condition. This aseptic condition provide in laminar airflow chamber. Here the inoculation means entry into the tube, into the culture tube or culture medium. This is carried out under aseptic condition in laminar airflow. After that, incubation under suitable condition for proper culture. The incubation period may be 5 to 6 weeks, may be taken 5 to 6 weeks. This incubation also carried out inside the laminar airflow. After incubation, some provide some callus like pot. After provide callus like pots, and this material subcultured again for obtaining desired result. After subculture, during subculture process, we apply some phytohormones also, extra phytohormones also, and vitamins phytohormones. As a result, the small plantlets are produced, plantlets are developed, which is finally developed as a plants. Now this plant transferred into the polythene bag and keep under the greenhouse for the weeks. After 5 to 6 weeks, now transferred into the outfield. So this is the techniques in tissue culture steps. Let's see once again. First of all, explant selection, remove of the extra parts, trim the explant. Now this is all sterilization. This line sterilization of explant, sterilization of glassware, sterilization with distilled water, and finally culture media sterilization. Actually, this culture medium sterilization before inoculation of the explant. Now actually the explant sterilized and culture media sterilized. Now these are ready to entry into the medium. But during time of entry into the culture medium, there is a chance to attack the microbes. If you done in outfield rather than laminar airflow, this activity, if you have done in outfield, there is a chance to attack microbes. That's why this inoculation process carried out inside the laminar airflow. Later, provide appropriate temperature, optimum temperature to incubation period of production of small callus. Again, subculturing to get a desired result. After plantlet form, transferred into the outfield. Before directly transferred to outfield, we actually maintain it under the. We should maintain under the palisade or greenhouses. So this is the tissue culture techniques. Next, tools and techniques plant tissue culture. Already tools autoclave. I don't want to waste your time and my time because there is the last time only 6 days remain. So already autoclave explained during practical training classes about laminar airflow also explained in training classes. I don't want to repeat again it. So I go through the next unit. Application of plant tissue culture is very important. Applications in plant tissue culture. Application of plant tissue culture. So actually, the plant tissue culture is being widely used for large-scale plant multiplication. Already told you earlier. Apart from its use as a tool of research, the plant tissue culture techniques have in recent years become of major industrial importance in the area of plant propagation, disease elimination, and plant improvement and production, and preparation of synthetic seed, secondary metabolites, and production of applied plants. There are the benefits of the tissue culture techniques, right? Let's discuss one by one. The first one, micropropagation. Micropropagation, at the plant, that means the number of plants are derived from the single plant. Number of plants derived from the single plant. For example, you selected desired plant. You selected from the desired plants more than one explant. For example, you selected 10 explants from the desired plant. These 10 explants inoculated into 10 culture media, incubated 10 culture media, provide optimum temperature to all culture medium. After 4 to 5 weeks, how many plants get developed? You taken 10 explants. So 10 plants are developed. Like so, if you take 100 explants, how many plants are developed? 100 plants are developed the same time. No need to take much time. The same time, here the 10 plants production same time, 100 plants production same time, 1000 plants take production same time. That is based on the explant. If you get more explant, more plants are developed, right? So that is the micropropagation. For example, here we take here the, for example, this is the donor plant. So this is the explant source. This explant source, as we discussed earlier, first culture media, this is the culture media tube, culture media prepared, sterilized culture media, explant selected, this explant collection, explant also sterilized. After that, this, for example, I selected one leaf as explant. If I select one leaf as a explant, only one plants are production. Like that, if you take the more than one leaf as explant, this inoculated into the number of tubes, culture media 1, culture media, second culture media. Here also, for example, this is the stage 1. The shoots are established in culture. From the explant, shoots are formed. These shoots, again, we subculture into multiple micro shoots. For example, here the just small portion formed. This small portion again subcultured into various medium. Finally, during subculturing process, apart from the shoot, root formation also formed. So root formation and shoot formation finally get the more plants like this and transfer it into the polythene bag and under the greenhouse. So this is the micro propagation. Here the how many plants are here? You have taken here only one plant taken. But one plant taken, you selected number of explant, you subculturing number of time, not only number of explant, of course number of explant also provide number of plants and also subculturing. This explant again you subculture, means from the one explant due to the subculturing provide more number of plantlets during subculturing and provide number of plants. Finally, we get more than one plants. This is called micropropagation. This is called micropropagation process. And another application of clonal variations. What is variations? How general variation occur? Where the variations takes place? Actually, variation takes place during reproductive cells during meiosis period, right? What happened during meiosis? What is first meiosis? Meiosis occurs in reproductive cells. During meiosis, the chromosome number decreasing off. While it is segregated into gametes, while this chromosome become off and segregated into chromosomes, there is this chance to formation variation in the form of crossing over. During crossing over, genetical recombination takes place. Genetical recombination. Due to the genetical recombination, variations occur. So these variations occurs in reproductive cells, not a somatic cells, not a vegetative cells. If this same type of variations occurs, that is called as a somaclonal variations. For example, in reproductive cells, chromosome. Let me change color. Actually, these are in reproductive cells. This is reproductive cell. This diploid condition. During formation meiosis, it divided, entered into the gametes. Now this diploid two together, diploid condition later becomes as a haploid after segregation. It is haploid. That means one chromosome here, one chromosome. Now become haploid. But during segregation, these two crossing over takes place. Crossing over means a bit of chromatid, here bit of chromatid cut and exchange their position, which means the red portion attached to blue color and blue color attached to red color. This is called recombination. Due to the recombination, variation occurs in coming generation, next generation. Variation occur in next generation. But in somatic cell, after formation zygote, diploid cell, zygote, only mitosis takes place, only mitosis. Mitosis means the chromosome number neither decrease nor increase or never formation, most probably not formation, any recombination, genetical recombination. But during tissue culture process, the variation may be formed. The variation may be formed by tissue culture cycling during subculturing process or during formation of somatic embryo. So this is occurred in vegetative cells, not reproductive. That's why called as a somaclonal variations. So this genetic variability is due to the cells of various ploidy of levels, maybe diploid, triploid, different type of ploidy. So this kind of variation may show some useful character such as resistance power to particular disease, herbicide resistance, test tolerance, etc. And some agronomical traits like tiller number, panicle size, flowering time, plant height, lot resistance, yield, nutrient content, and different kinds of morphological variants in leaf. Actually, this is occur due to the some variations. For example, you take the neem tree. This is neem tree. Neem tree, for example, for our better understanding, the chromosome may be this like. Only this blue color type of chromosome present in the neem tree, this is maybe not resistant to disease. When you alter the genes during tissue culturing, so the same gene altered as some like this. Maybe alter like some changes takes place during culturing process. So as it changed the genome, it reflects Some desired characters may be herbicide resistance, stress tolerance, and also they may be the show in some morphological characters also. That means panicle size may be decrease or increase, flowering time also may be different, plant it is effect on plants height also, and also production of the yield production also. So these are done by somoclonal variations. Next one, secondary metabolites. First of all, how primary metabolites form? The primary metabolites are formed during photosynthesis. Photosynthesis, carbohydrates formed. After carbohydrates formation, these are utilized in the body parts. Some of the excess of carbohydrates, excess of protein stored. Actually, in what happened in animal, the primary production after utilized, the secondary production are leaved out in the form of urine and desiccation, urine or desiccation process. That is done in animals through excretory system. But does the plant have excretory system? No. As the plants not have excretory system, the all waste material, the waste material formed after utilization of the primary production, primary material, all primary material utilized, the end result of primary production, utilization, secondary production, secondary metabolites form. As lack of the excretory system, the secondary production stored inside the plant body. They are secondary metabolites, such as alkaloids, maybe alkaloids, flavonoids, different steroids, there are. So like that, we can do by tissue culture techniques, production useful compounds, useful secondary metabolites such as like alkaloids, quinine, steroids, glycosidic compounds, essential oils, coloring agents can be done by tissue culture. So this aim can be achieved by selection of specific cells, producing high amount of desired compounds and development of suitable medium. For example, we need one alkaloid. So vincristin, which is obtained from Catharanthus plant, right? So we selected excellent Catharanthus plant. After culturing Catharanthus plant, we can get only these alkaloid plants, more and more alkaloid plants, like the same quinine also. And for example, another one is not given. Plant cell culture rather small amount. Okay. For example, so mainly vincristin, in vincristin amount more in leaf rather than seed or stem. For suppose this is Catharanthus plant. Catharanthus plant have the stem, root, leaf, but the amount of vincristin only the net amount, that means highest amount present in leaf only. First of all, you separated the leaf. From the leaf, you separated the tissues where the secondary metabolites formed. You selected that tissue only that tissue only, rather than root and stem and flower and seed and fruit. Only leaf. Inside the leaf, where the tissues are deposited, the secondary metabolite tissues are deposited in the form of increstin. You trimmed that particular plant, that particular portion only. That is would be explant. Now selected explant. This explant after sterilization inoculated into culture media. We know already how to prepare culture media. After inoculation, incubation in the laminar airflow in laboratory. After weeks, it provides the plants, same vincristine plants, but which has more and more vincristine, secondary metabolites. Like that, vincristine, so this act as an anti-cancer. Based on that, we can prepare the drugs, anti-cancer drugs also. And also we can prepare the quinine, which prevent the malaria fever. Like so, we can prepare the number of alkaloids and glycosidic compounds also. Right, so this is the matter of secondary metabolites. And another one is haploid plants, production of haploid plants. Production of haploid plants. How we get haploid plants? Haploid plants production from the haploid chromosomes, haploid part of the plant. In plants, generally diploid condition, but in flower after meiosis, haploid pollen grains are formed. So like this here, in place of explant, we selected the pollen grain. Selected the pollen grain, pollen grain, pollen grain as an explant. The pollen grain always applied. This pollen grain or ovule also we can select. Ovules also we can select it. So for example, if you take pollen grains as it applied, the pollen grains inoculate into culture media. In culture media, what happened? Callus formation, subculturing the callus. As a result, finally we get the plant. Now the ploidy. Actually, this is, for example, say we take neem tree. Neem tree. Do you want to the neem tree in applied condition? What we select instead of vegetative parts, we selected the pollen grains, haploid pollen grains of the neem. The neem pollen grain after culturing, production of the neem plant, the ploidy of neem plant here haploid only because there is no fusion. Directly haploid condition. Haploid condition get developed into haploid plant. If with the same polyploidy condition takes, polyploidy plants for plants also develop. Right. Many of these recessive traits can be made by expressed in double haploid, such as low glucosinolate content in brassica. For example, in brassica. Right. Next, actually forgot soma clonal variation, right? Somatic embryogenesis. Forgot somatic embryo where it is. Somatic embryo formation, somatic hybrids are somatic embryogenesis. Right. Somatic embryogenesis. How embryo formed in general? Hello? How embryo formed? Embryo formation, male gamete, female gamete. If the male and female gamete fuse, as a result, formation of zygote, it is diploid. Zygote. This zygote later developed into embryo. This zygote developed into embryo. This is generally embryo formation. But during culturing, plant tissue culture, embryo formation, these embryo called as somatic embryos. The process called as somatic embryogenesis. By direct explant. So from this embryo, we can store this embryo in the form of synthetic seed. Synthetic seed, somatic embryo are nothing but called as synthetic seed. Synthetic seed. After encapsulated with alginate. After formation embryos for future use, actually for future use, we store the seeds in general, right? But after formation embryos, some somatic embryos encapsulated with the help of alginate, sodium calcium alginate layer. Now this encapsulated embryo called as synthetic seed. We can store these synthetic seeds for future use also, right? As the somatic embryos are derived from the single cell, not involved in two more gametes, not by only single cell, this method is very much useful for production of disease-free propagule. Why? Because, for example, this is why this male gamete from one plant, female gamete from one plant. Already, this production of this male plant already infected. Maybe if infected, what happened after formation zygote? The zygote, embryo formation, later seed development. This seed produced as a plant, produced as a plant. But here, this is the female plant or healthy plant. Female plants which is produced the female gamete which is healthy, but not male. But after fusion, what happened? The deceased plant obtained. But during somatic embryogenesis, that means during formation of embryo, somatic embryo, actually with this embryo formation without fusion of any gametes, right? Maybe you selected the only female plants. Already you know that female plants are well resistance power. It has well resistance power. Already there are two plants. One is male plant and female plant. The male plants are already infected with disease. In the situation